rationally choose the best natural ecosystem to survey metagenomes to discover novel antibiotic or the natural product. So the idea is like, suppose if you want to discover a particular class of antibiotic, can you have a rationale that, okay, you go to such and such longitude and latitude location on the earth, and you will be able to, there are high chances that you might actually recover such kind of clusters. So can such rationale be developed? And like this question has been formulated like, has the world profiled enough metagenomes for natural product discovery, or serendipity is the best bet yet? So we know the answer to this question, that all the discoveries with respect to sampling are actually serendipitous discoveries. And unless and until we capture maybe out of 1 trillion microbial species, at least say 20 or 30% of that, all the discoveries, according to me, will be serendipitous discoveries. And Professor Jansen from University of California has also given a thought and has actually formulated some outstanding questions like, at what rate are the biosynthetic gene clusters created and lost? So when you have huge amount of data of metagenomes, you can probably answer such kind of questions. And how often do the new chemical scaffolds evolve? So again, this the answers to this question lies in the big data. So with this, I have come to the end of my talk, and I would like to acknowledge the groups at Tübingen as well as at the institute where I am currently. We are actually having a very good computational facility which has been supported by Ministry of Science and Technology. This is a national Param supercomputing facility, and it has kind of 850 teraflop of computational capacity, and the universities and institutes which are in the Chandigarh and Mohali regions are actually using it. And if any one of you would like to actually use this facility, you can get in touch with us. The CDAC has actually given a scheme where they are giving 1 lakh rupees worth of computation free to a professor, and 50,000 rupees worth of computation free to PhD student, and likewise for bachelor's and master's students. So if in your research, if you happen to use supercomputing, then we can actually help you to run your jobs on our cluster. So with this, I would like to thank you for your attention, and we'll be happy to take up your questions. Thank you.
Thank you, Dr. Mantri, for such an excellent talk. Now it is open for the questions.
Hello.
Yeah.
Sir, good evening, sir. Nice to hear your talk. So myself, Tulasi from Naipur, Hyderabad. So I'm working with the fecal microbiota genomic samples.
Right.
So like, as you have explained the difference with 16 sequencing with the amplicon as well. So my question is like, will it be sufficient to establish the primary data if you're going with the primary metagenomic analysis? As in bacteria, we look for only targeted like V3, V4 regions. So is it like fine, we have to establish both, or is it like one will be primary enough?
Yeah, because the diversity which is present in gut samples is less as compared to what is present out there in soils. So in that case, yeah, 16S would be sufficient. And but if your goal is not to just study the diversity, but to also check which biomolecules are formed by the microorganisms which are present in the gut, then of course 16S won't be sufficient. So you will have to actually go for shotgun sequencing. And the patterns which you will see would be slightly different in case of gut microbiome. But in case of soil, like I showed, that the patterns of taxonomic diversity are completely different, and you should rely more on shotgun data rather than 16S data.
Yes, sir. Like only to understand the taxonomic profile or species abundance, like this will be fine, I think.
Yeah, yeah. For gut, I think it will be fine.
Yes. Thank you, sir.
Anyone else?
Dr. Mantri, this is a great opportunity that you people have got experience in genomics and other related data, and we are mostly into modeling,
Yeah.
and we are just neighbors.
Yeah, of course.
So our exchange of views will be of great value. Many of the NIPER students, if you don't know, you better visit Nabi. Nabi
Yeah.
is just
Yeah.
few meters away from our
Exactly.
place, and they have got excellent research opportunity in terms of food and nutrition. So we are working on many projects. Right now, many of you may know that we are working in collaboration with National Institute of Nutrition in Hyderabad. Why to go that far, Hyderabad? Why not here?
Yeah, of
So
course.
only thing that when we started, Dr. Mantri was not in town.
Yeah.
He went for Germany, so we needed help from Hyderabad. So we are working on nutrition also. So when we work on drugs and nutrients, certainly there is a lot of learning to be done from the agrobiochemical perspective. And his lecture today has thrown opportunities to us and helped us in realizing what can be done. And they have established excellent facilities. I was part of it initially when they were establishing in the committees and others. So nice talking. Thank you for giving very nice lecture.
In continuation to what Dr. Bhatam said, we would also like to have the data, like the access. Can we have the access to the data to use for some modeling or something else? Something like the collaboration. That will be like a good collaboration we can have.
Yeah.
The data you are generating, lots of data, and if we can use it for the modeling work, maybe
Yeah,
someday.
I think in case of metagenomes, we haven't generated a lot of shotgun metagenomic data set. We have some plans, but in public domain there is tremendous amount of data. So, and we actually are kind of developing pipelines so that we can harness the power of the computing facility which we have to crunch all that big data which is available in public domain, so that we can have databases of, say, biosynthetic gene clusters, or databases of genomes of all the microbiomes. So such projects we have initiated after I came back, and those are actually now in pipeline. But in terms of computation also we are rich, and in terms of storage space also we are rich. So for the cluster which we have around 1 petabyte of storage, that is 1,000 terabytes. So if you have any big data which you want to crunch, we can actually house that big data, and then you can write your programs to crunch that data. Yeah.
Thank you.
Yes, we come to the end of this session because one of the speaker could not make it to this dais. He became busy with something else. So now we have got time and sufficient time to discuss about the topic of concern. This is pharmacoinformatics. We are going to have a session. Okay, please. So, Dr. Mantri. We have got sufficient time now. The topic of concern is informatics. Informatics science is growing extensively, and the pharmaceutical sciences is also growing extensively, especially after this COVID impact. The importance of the pharmaceutical scientists for the world and his services have been realized by many administrative officers and the government. So as a result, there is a recognition, and we need to be able to make best use of this situation and contribute positively for the growth of both the fields. Now, are they separate fields? Are they integrated? Yes, somewhere in 2002, long time ago, exactly 20 years ago, we conducted a small conference called Pharmacoinformatics. At that time, we thought informatics and pharmaceutical sciences. But then it was discussed, pharma is not any sustainable word across any science, all scientific community. Pharmacon means drug, and pharmacoinformatics, that's how pharmacoinformatics it became. And later on, our Board of Governors decided that we should encourage this topic in 2002. That was good news. We felt very happy that our institute is taking leadership in the field of integrating informatics and pharmaceutical sciences. That is what all we are going to discuss. Before that, I invite a few panelists. Professor Wade, please. Professor Sophia. We are very lucky to have Professor Ravichandran ji here, sir, as a panelist. Professor Garg. Dr. Balwinder Singh. He was busy and he said he has to leave. Dr. Mantri. Please take seats, please. I will be coordinating, so I will be moving around. I will not take. Please take seats. We have one guest from Naipar Guwahati, Dr. Dikshit. So I am telling that what happened 20 years ago in our institute. Our director asked, what can we do? We said, sir, maybe we can conduct a certificate course. Please give us some money, some 50,000. He said, no, I will not allow certificate course. Then we asked, sir, we can do some diploma course. He said, no, PG diploma and all this, there is no value for such. We asked 15 lakhs for that. He said, no, I will not allow all that. I will only give you 1 crore rupees if you want to do something big. If small thing, don't do small things. Do big things. So certificate course, not to start a PG course, pharmaceutical sciences, masters in pharmaceutical informatics. If you can do, you do it. Otherwise, I will use this money for something else. So that was a big challenge. We took that challenge and we started this department called Pharmacoinformatics. So slowly we were trying to understand what is this topic, how we can use. We started with molecular modeling. Molecular modeling using quantum chemistry, then slightly bigger molecules of quantum chemistry, complete drug molecules. Like that we started slowly, and then we expanded in all directions of computer-aided drug design, that is virtual screening, pharmacophore mapping, molecular docking, and all the technologies that were emerging at that time. At that time they were emerging, even today I am sure they are still emerging. They have taken a big shape, no doubt. It has taken a significant improvements have been made. But as far as data curation and the value that we want to attach to the data perspective and analysis. India is yet to learn the highest standards which are being adopted across the world. So that is the background in which we would like to start discussion. Our head of the Department of Pharmacoinformatics is here, Professor Prabha Garg. Please say a few things about the current status of the pharmacoinformatics and future perspectives.
Regarding the pharmacoinformatics, whatever anybody says, but you have to believe in yourself. This field is the coming field, the data science, the informatics, analysis, computer-aided design. Even if you see number of scientists who are not from the computer science, like the chemistry or the physics or the biology, they are recruiting the more of the computer scientists in their labs for the more of the analysis of the data. So have faith in you, believe in yourself, you can do number of things with the analysis of the data by doing any one of the approach, not only the one approach, the artificial intelligence or the computer-aided drug design. If you know the number of techniques, you can just make the combination of this, whatever you need to extract the information or the data, try to apply that. And I'm sure tomorrow is the, for the pharmacoinformatics more than any other department for the drug discovery.
Ravinder Singh has joined. Please come to the dais. Okay. Dr. Dixit. I will be communicating between the students and the panelists, so please continue. Professor Balwinder Singh has to leave quickly, so comments on the future course of this field of computer-aided methods integrated with pharmaceutical sciences.
Yeah, thank you, Dr. Bhartam. Actually, I mean, the field is kind of a wide open. The informatics, data science is coming up like anything. AI is playing a role, I think, in every part of our life. Now, further, the big boom which has come up in terms of data, as Dr. Mantri was saying, that we have a lot of genomics data in our hand. Though this data is available in public also, and data is also being generated in our labs. Though we are talking about, microbiome data, though we are talking about any metagenomics coming from, soil samples, gut samples and things like that. So a lot of data is being generated. The thing is that we should be ready to, have the tools like this data, and I'm sure will lead to many more things. And definitely, it has been years since the last antibiotic came. We definitely require antimicrobial research, or the AMR antimicrobial resistance is coming up like anything. So we definitely need more data analytical techniques to address the problems in the genomics, and then try to look at the metabolites secreted by these microorganisms, and coming from metagenomic approaches, from any other approaches, whereby how these can be exploited in terms of used as an antibiotic, used as for any kind of health or other fields for that matter. So future is kind of a wide open, and definitely AI will have a great role to play in the coming days, and so is I think the informatics and pharmacoinformatics, because new drugs will come, these secondary metabolites are being secreted. We have to look at their functions, mechanisms, and look at their variety of properties that they can hold. So like this, I mean, this field will have wide ramification in other areas, health, agriculture, and water, soil, everywhere, for that matter. Okay, thank you.
Professor Sobhia is an expert in bioinformatics. She has been teaching to the students for the past many years. She is bridging the gap between the knowledge that we have in biosciences versus the informatics science. So she will give comments.
Good evening, everyone. So I work in the area of proteins and the structure-based drug design. So I can just give you some information. So the first thing what I want to say is, so we are all called as in silicoids. So the work what we do, so they are all virtual molecules. We call them as hits. So, but that has to be translated. So we face a lot of challenges in that domain. So we finish it. So we all have more than 100 publications. We get novel ideas, some innovative thoughts, and we just publish them and finish it. But there are challenges where we are not able to take the molecules further. So that is one problem. So as students, I will tell so we have to be aware that whatever we do, we have to do it carefully and with the presence of mind and what we are doing. Otherwise, it will go like GIGO input and GIGO output. So if you give, so whatever we are doing, so it is we who control it. But on the other side, there are a lot of scope in this area, and it has been again and again proved. So we couldn't have actually many speakers in this session. So, what I want to say is there are many challenges, we can solve it. And pharmacoinformatics, one of the areas is finding out molecule, and we should contribute. We have all the facilities, we have minds, we have brains with us, and I expect, including me, that something we should do, and we should get some molecule to some extent, being from Pharmacoinformatics from National Institute of Pharmaceutical Education and Research, Mohali. So that we can do, I have great hope that we can do. So whatever, so I want to encourage all the students, whatever limited infrastructure, whatever challenges we have, but still we can do. So sometimes we have to fight, sometimes we have to shout, we have chilana hai to get our things, but we can do. There is a lot of scope. Recently somebody was asking the question on AlphaFold. Yes, artificial intelligence, molecular dynamics, but whatever you do, please don't forget the basic things. If we miss the problem, why we are working on it, so we may lose many things. So I want to encourage all of us who are working in this area that we can contribute something if we do with all our heart and what we are doing, knowing, and if we do it. So I'll just give it to the next.
Yes, Professor Ravichandran, he is a leader and he is guiding an institute where pharmacoinformatics has been given sufficient importance, and he has been guiding many leaders already. So I wish to listen from his.
Thank you, Professor Bharatam. Basically, I am thankful to all the speakers of the evening session. The pharmacoinformatics, it is a big area. So, how it is organized manner in the application, that's a very important component in the, like it is equivalent to big data, how it is important for that. Several other profession, the big data role on this. So what I have here that in the pharmaceutical in the sense, in the pharmaceutical formulation is a very important component for which how is the data on excipients, the choosing of the data and excipients are the very important role for that bioavailability, bioequivalence. This is one of the very important area while we have several off-patented drugs are currently it is there as a challenge. There is no much takers on this, how the pharmacoinformatics data can be useful, software can be useful for that identifying equivalence, in mainly focusing on that excipients, selection of the excipients. So this is one of the aspect. Number two, what I can suggest in the proteomics, what the Professor Balwinder Singh, he was saying genomics, the colleague also, he presented that on genomics. Mainly in the proteomics component, it is a schematic role in the all aspect of expression. So what the proteomics expression, any change on that, the mutation, epigenetic modification, genomic changes, it comes to that end with the proteomics expression, modification in the proteomics function, protein function, whether it is a upregulation or downregulation of the proteins. These are the very important component. How this the data can influence with that the different kind of finalization on mainly biomarker. In the theranostic, either it is a diagnostic or in the specific part of that for identifying multiple protein for that the new pharmacophore identification. By this way, there is no problem with only one protein. There are more than one protein. It is how you can confirm that you have to get the data of single problem. What are the different protein is expressed with the different proteins, how that the existing drugs works on that. And another component is that epigenetic modification. The pharmaceutical on epigenetic modification, it is a preventive measure. The preventive measure is the very important for that any healthcare management. The preventive measure, how you can have, if you develop the data on that, you produce the data for this purpose, it creates the evidence on that, how these the pharmaceutical, any existing molecule or the new molecule, how it works on the system, epigenetic modification and prevention aspect. So these are the basic component. It includes that. I don't want to repeat whatever he presented. It is a wonderful data on that, the genomic component. It is a very important for that, any basic aspect of genome to that the protein expression. These are the logically, the component need to be integrated and presented well. So for which design is very important. For the Professor Bardam, what he is doing, he is trying to pull that, though I am not the person with that the informatics aspect, we conduct the course program, I don't say that no on that, so, but still, the conceptual wise, how this can be strengthened. So we conduct a couple of the projects also in the rare disease. The rare disease, you all know that the problem of, genetical disorders, it comes into that the rare disease. There are several thousand. There is as per the record currently 7,600 plus rare diseases are there, but there is no expertise on this. There is no diagnosis for this purpose. But how this available data, it suits with this kind of the problem. This is one of the important aspects. While we go for several challenges and demand on that. What the several challenges? Pharmacoinformatics, how we can involve on the bacterial resistance. Antimicrobial resistance is a major challenge in everywhere. It is not only bacterial resistance. Now it has emerged with the viral resistance, fungal resistance, cancer resistance, entire things it get resisted. So how this the data will be useful for that the selection of that the material, it's a AI-based drug selection, prescription. These are the components are very, very important. Now it is already the AI-based molecular selection, how it is used.